Okay, hello everybody. Welcome to our first talk in the Phylogeography, a Growing Bridge Symposia. I'm very pleased to introduce to our keynote speaker, Dr. Fernanda Werneck. She works in the Instituto Nacional de Pesquisas de Amazonia, and she's now going to talk about narrowing the adaptive bridge in phylogeography, history and future of neotropical herpetofauna in changing environments. So please, Fernanda, if you want to start your talk.
Hello, Lara and everyone. Let me just share the screen. Lara, can you just double check with me if it's fine and all smooth on the screen?
You're good.
Please.
Yeah, it's okay.
Yeah, it's good. All right. All right. Good afternoon, everyone. It's a real pleasure to be here today sharing this really nice event. I'm talking from Brazil with you. I've always been very advocate of diverse events and research groups. So it's really a pleasure to participate in the Biodiversity Research and Researchers Diversity SBE 2021. Oops, I think I should try and take this off here. So here we go. So today I'm gonna talk a little bit with you about how trait-based phylogeography and ecological modeling, coupled with local adaptation studies along large environmental gradients, can contribute to understand biodiversity patterns and species, and possible species responses to environmental change at the megadiverse Neotropics, with a special focus on the herpetofauna. I'm a herpetologist myself, and on contrasting environments in South America. So natural selection and local adaptation along these large-scale environmental gradients can play a central role in generating biodiversity. Yet, the integration of local adaptation into phylogeographic inference has been largely underexplored, mostly due to the early emphasis of the field on the neutral genetic variation. So integration of population genomics and spatial analysis can provide us the opportunity to detect environmental selection on traits that are relevant in phylogeographic and ecological contexts, such as physiological traits, for example. And we can do that using several sorts of approaches. So for example, we can use bottom-up approaches such as common garden experiments and transcriptomics, that we start off with the definition of putatively adaptive genes, and then we move on linking the variant alleles of these genes with phenotypes or the environment. As we can probably imagine, this is usually quite a challenge for natural populations of non-model organisms, such as the ones I work. So for these cases, we can also use more like top-down approaches that help us quantify the geographic variation of relevant traits, and then identify associations between the environment and adaptive genetic variation from a large sample of random markers, for example. So given that we know that diversity patterns, they can correlate with environment and climate across multiple geographic scales, it's sort of logical to expect that then when the environment is going to change in space and time, then the associated biodiversity and natural populations might also change, right? And in fact, in response to climate change, species can have two groups of main natural compensatory responses. So they can first move to track down the favorable climates, and then with time go through some range shifts that can happen along latitudinal or altitudinal axis, and also across deeper or more shallow recent timescales. When we talk about these deeper, more biogeographic type of timescales, phylo-climatic approaches that sort of integrate niche models into phylogenies that can help us understand patterns of niche evolution and range shifts across large scales, or questions such as whether the niches are conserved or if they change over time, and if that can facilitate, for example, biome shifts across biological clades. Meanwhile, at more recent time scales, the species might also experience faster and more significant changes in distribution in response to all those strong environmental changes that have been induced by human action, right? So understanding these range shift responses and dynamics across space and time, it's very important, not only because then we can unravel the historical causes of speciation at phylogeographic relevant contexts, but also because then we can also incorporate this information into how biodiversity patterns might be affected by future climate change. Yet those sorts of studies are very rare here in the Neotropics. So that was sort of the first class of responses, but on the other hand, besides range shifts, species can also go through adjustments to the new environmental conditions that they might be exposed. And that can happen thanks to phenotypic plasticity or evolutionary adaptation, when we can actually detect signatures of local adaptation and natural selection by investigating adaptive genetic variation. So quantifying these relative roles of range shifts and possible local adaptation or evolutionary responses in meaningful ways that can inform us about population level processes, it's quite a challenge, but it's also an opportunity to understand how all this interplays on the speciation processes, species distribution, and of course, species vulnerability as well to changing climates. So talking a little bit about ways that we can quantify range shifts in this population sort of information that's meaningful for us. So they're using ecological niche models, right? So basic correlative models that use current distributions of the species as a proxy to relate them to the environmental variation, they are very much used, even though they, of course, have a number of simplifying assumptions. So another class of ecological niche modeling is the so-called mechanistic models. They, instead of just relying on occurrence data, they can use biophysical properties of the populations. For example, physiological data of temperature preferences or critical thermal limits, to then link more directly functional characteristics of the species to the environmental conditions, and those can also be projected back into the geographical space and make this functional link more explicit. So as for the evolutionary responses, the Genome Environmental Association studies, they have emerged as a very useful tool to search for candidate SNPs that might be under selection and provide us signatures of local adaptation in mediating these diversity patterns and structure, and also species responses, right? So the GEA, the Genome Environmental Association studies, they kind of connect population level genomic data with local climates along environmental gradients, and then go on to identify the standing genetic variation that might have evolved under selection. And they can be very important because then this information can be integrated into assessments of climate change vulnerability, for example, and bring us insights into how climate change adaptation across natural populations and phylogeographically relevant lineages can improve our understanding of the potential impacts on species distributions to their vulnerability to climate change. So in general, studies that have been incorporating information on physiology, trait variation, and local adaptation to climate change assessment, they have shown that by incorporating these information, we tend to recover less extreme range loss projections in the future. So here are a couple of examples of mechanistic modeling studies showing this pattern, and also studies that look at for signatures of local adaptation, showing that using this information decrease these future range loss projections and can give us more realistic scenarios of how populations might be affected with changing environments. And of course, the importance of understanding that the interplay of these characteristics in species response is that if they fail to go through any of these adjustments, then they might experience populational collapses that can ultimately cause local extinctions and even across the entire species range. So as an example that is relevant to the area that I'm going to talk about, I'm talking to you today, Sinerva and collaborators in 2010, they have used mechanistic modeling, integrating thermophysiology data of lizards with future climate change projections to calculate a metric called restriction hours, which would be the number of hours of the day in the future that environmental temperatures would be higher than what those populations might support, and then that would cause individuals to restrict their activities by the excessive heat in the environment. So using this approach, they have modeled large areas of high extinction risks for lizards globally, and particularly in the South America. At that time, the study didn't have a lot of field validation in South America, and that's sort of where I'm getting into today to you. But the thing is that that's not only shown by these studies. Several other studies, they have proposed that amphibians and reptiles in South America are particularly vulnerable to the impacts of climate change. And of course, this is, as I said, not only for these groups, but globally, where we are going through this very extreme biodiversity crisis, in which up to 25% of the species are threatened and more than 1 million are under some sort of imminent extinction risk. And those are numbers of 2019, right? And that brings me to the Neotropical region in Brazil, which is considered the #1 megadiverse country on Earth, in one of the most biodiverse regions, the Neotropics. And the continent spans a huge latitudinal extent, with very dynamic climatic, geomorphological, and paleoenvironmental setups, and harboring this huge biodiversity in contrasting environments that are usually in very narrow contact, such as the very large rainforest savannas transition that occurs in the gradient between Amazonia and Cerrado, and I will talk in a minute about them. So that's sort of an ideal environment to kind of integrate phylogeography level information and understand signatures of local adaptation across large environmental gradients. So in 2011, Antonelli and collaborators, they used a broad meta-analysis that showed very high levels of biotic exchange among these large neotropical biomes or ecosystems. And they recover very frequent biome shifts across biological clades between major vegetation types. And the shifts, they were mostly recovered from the forest ecosystems into the open biomes. And that sort of prompted them to propose that the Amazonia is the primary source of neotropical biodiversity. So that also have left open that question whether this biotic interchange between these biomes, they would be resultant from neutral random process that just accumulate over time, or if they were triggered by adaptive innovations that allowed colonization of these new environments. And although South America is a nice natural experiment for neutral biogeography and adaptation investigation, we have virtually no study on the search for loci, for example, under adaptation on natural population across large gradients. So just to show to some of you, I'm sure several of the people here are from the Neotropics, but others are not. So here, just placing you a little bit in this environmental context that I'm talking today. So as you can see, we have very beautiful landscapes in contrasting environment, but not everything is just beautiful landscapes. In the next decade, threats have been escalating a lot across Brazil and there are many other regions, but the main focus here in my talk, it's the transition between the Amazonia and Cerrado, which actually largely coincides with the region where most of the deforestation in the Amazonia is taking place, or the so-called deforestation arc. And of course, this is very interconnected with the political scenario that we are on, that are increasing, rapidly increasing all the threats to biodiversity and ecosystems conservation with many synergies between deforestation and climate change going on. So just as a brief example, recent studies, they have advocate that both the Cerrado, or the large savanna, and the Amazonia rainforest, they are very close to their tipping points in terms of plant species extinction in the Cerrado and non-forest conversion in Amazonia. So that kind of brings a sense of urgency to understand the interplay of impacts and past history and how we can integrate them to understand the future of our lineages, right? Our systems that we study. So as I said, I am a herpetologist, and before diving into some of the case studies of my lab, I just want to briefly bring some facts for us to keep in mind along the top for neotropical herpetofauna, that basically most of the biodiversity shortfalls, they are applicable for amphibians and reptiles in the neotropical, which means that we know relatively little about species limits, their distributions, relationships, and even less about their tolerance to the environment. And the consequence is that as we do those sorts of population genomics studies, we have a strong need to refine data at the interspecific and population level through lots of field work and use and strengthening of biological collections, which is what we do in my group. So here, just a few photos that I bring. I usually like to show more, but it's a short time. So it's just a point that I want to make, that all the work we do, it's integrated between the field work and biological collections. And as side products, we always have new species discovery, checklist, community ecology studies as well, until we get to the point of actually getting into these sorts of questions that we are talking today. So that brings me to the studies that we're going to dive in to understand the interplay between neutral and adaptive ecological factors on the speciation or phylogeography patterns and species response at distinct temporal and spatial scales. So today I'm going to talk about three main studies in my lab. The first one, it's a phylo-climatic study with a genus Kentropyx. Second, we're going to dive into a trait differentiation and mechanistic modeling for one of the species that we also have phylogeography information. And finally, for the same species, it's an adaptation, climate adaptation study in which we want to identify signatures of local climate adapted genetic variation. So as for these studies that I selected today, they are all with a lizard system. I'm going to briefly present the system to you. So the Kentropyx genus is a very distinct genus within its family, which is the Teid family. And the genus as a whole has a broad distribution in South America, occurring both in the dry diagonal biomes and in the rainforest, both the Amazon and the Atlantic forest. But the genus has some species groups that have more restricted distributions. So three species group. The first species group is the calcarata group that has 3 species that occur either in the Amazon or in the Atlantic forest in the case of calcarata. And morphologically, they are very distinct from the others by having very small dorsal scales that are very easily distinguishable by from the tail scales. The second group, it's the paulensis group that has 4 described species and a couple of other undescribed species that were recognized based on morphology, variation, and genetics as well, but they were not described yet. And this group is distributed mostly in the open ecosystems of the Brazilian Shield, with 2 of the species actually being endemic to the Cerrado, and the other 2 also occur in the Chaco-Paraná basin, more flooded type of savannas. So the dorsal condition of these species, of the scales, they start off small, close to the head, but they tend to increase in size towards the tail, where they are nearly indistinguishable from the tail scales. And finally, the third group is the striata group that occur in the open ecosystems of the Guiana Shield, northern of the Amazon basin, and in some Caribbean islands as well. So this group has like large plate-like scales in the dorsal scales that are not different from the ones in the tail. So why I'm saying that is that because studies from the 90s, they have proposed that the dorsal scales, they increased in size during the evolution of the genus, with calcarata, paulensis, and striata group in disorder, kind of arranged in a linear progression of the size of these scales, and which at the time was proposed as an adaptation to thermoregulation and control of desiccation to drier climates in the Guiana Shield. And if that was true, then the genus would have evolved from the forest formation open of South America. However, in the day, that was my undergrad research actually, we proposed that just make sure I'm on, because I got a message that my internet is unstable. Lara,
No,
you there?
Okay. You are on. Don't worry.
Okay, all right, thank you. It's just like this, making sure that I'm not speaking with myself. So in that study, we actually proposed that we recover relationships in biogeography in ferrets that proposed that the diversification of the group might actually have occurred from the open formation into the forest formations. But at that time, it was only mitochondrial DNA based, this study, and we used just simple parametric biogeographic reconstructions. So gaining leverage of new data, multi-locus data, and phylogeographic approaches that we use to describe the niches of the species, we want to readdress this question and then evaluate the role of niche evolution on the diversification of this genus of lizards. So that was part of Yomi Shell's master's in my lab. She defended a few years back. In the study, it's already out. And then that's the sampling. We had 5 markers, nuclear and protein coding, mitochondrial protein coding nuclear loci, and a very dense sampling across all the species and all their distributions. So using this data, we inferred a dated species tree, which recovered the 3 species group clades and proposed that they mostly diverged during a middle Miocene. And the biogeographic reconstruction that we recovered indicated a role of susceptive dispersal events from an ancestral range that covered both Western Amazonia and the Brazilian Shield. And that would be sort of different from the large study of Antonelli that proposed that most of the shifts would go across clades would go out of the Amazon only. So then we recovered this subsequent dispersal from Western into Eastern Amazon and just more recently into the Atlantic forest and the Chaco-Paraná basin. So we went on, inferred the ecological niches for all the species their profiles. So we did that for several variables, and these are just some that I selected to show here. And by reconstructing, by making ancestral reconstruction of the species tolerances across the tree, so this is the same tree that you see in the left, and also tests of niche overlap and niche diversification between species, we found that most of the clade that is in the Amazon, both in the basin and the Guiana Shield, a signature of niche conservatism. But when we look at the clade that is mostly in Brasilia Shield, we found a very striking signature of niche divergence. So you see that this clade, which is corresponding to this one, the mostly blue and purple here, it occupies a very different ecological space when compared to the other two species group. So that's the graphical abstract of the integrative approach that we use between species tree, niche occupancy, and the ancestral reconstructions, and prompted us to conclude that dispersal and climatic-driven divergence likely resulted in adaptation to these new environmental niches and helped promoting habitat shifts and shape speciation patterns across this group, suggesting that ecological divergence might have a more important role in the diversification than previously thought, and then only neutral models would suggest. So then we moved on for a phylogeography study of one of the species focused mostly on the eastern Amazonia, which is Kentropyx calcarata. And we were interested in, which was part of Aureus Cronenberg Masters in collaboration with Museu Paraense Emílio Guild. And we were interested in analyze the role of the Amazonian rivers in structuring the population structure and in phylogeographic lineages of the species, which is a fairly common and species with a relatively high dispersal capacity. And what we recovered was that the species has 10 very well, mostly well-structured, deep structured lineages that diverged in the transition between Miocene and Pliocene, and they are mostly limited by the main Amazonian rivers. However, the correspondence is not 100%, as there is some haplotype sharing across some of these river margins. And we discuss in this study how the combined influence of rivers and climatic fluctuation have driven the diversification of these species in eastern Amazonia. But we were also interested in asking whether these lineages would have any signature of local adaptation of trait differentiation about these phylogeographically relevant lineages. So to investigate that and integrate trait variation and possible effects of environmental selection at the population level of Kentropyx calcarata into predictions of climate change, we have the study of Emerson Pontes that finished also a few years ago in my lab, his master's. And we want to evaluate those questions by investigating how thermal biology vary throughout the species geographic ranges and the consequences of it for lineages vulnerability under changing scenarios. So going into the 10 lineages that we recover in the phylogeographic study, we were actually able to sample 4 of them, sample for the physiological experiments. They are very intensive to conduct. So we did these physiological tests to estimate preferential temperatures, thermal tolerances, which are the minimum and maximum critical limits of the species. Oops, let me play here. And the sensitivity of their local performances under higher temperatures. And we also quantify operational environmental temperatures. So all these variables, they then were integrated to calculate the restriction hours, as I defined earlier. And under a mechanistic approach, we were able to make some predictions of suitability and distribution in different time frames for Kentropyx calcarata. So here is the main result of this study. We have others, but that's the main. And the way that the maps should be interpreted is as the distribution of the environmental suitability for the species. We actually model the lineages separately, and we are just plotting them together here. But the cooler colors are the ones with more favorable climates. And the hot color are the areas with lower suitability. So the difference between time frames can be interpreted as can represent the loss or the gain of adaptability. In the case of the loss of adaptability, that would be a way to infer local extinction risks. So as we can see here, our results show that there is a lot of variation in susceptibility across lineages. So some of them, they actually are likely to be more susceptible than others, to have higher extinction risks. Mostly, you see the Eastern Amazonia, where it's very cooler here and way more hotter colors in 2070, which is when we projected. While other areas, they actually might gain adaptability, such as a little higher altitudinal areas in Roraima state, for example. So that shows us that the phylogeographic lineages of these species, they have vulnerabilities that can vary across geography. So extinction risks, they are structured and their vulnerability, they will change from place to place. And also when we compare the thermal traits, we found that the populations that are close to the Amazonia Cerrado ecotone, they have higher and less variable maximum critical limits. And that's kind of, together with other studies from the group, suggested us that maybe these populations that are closer to the border of the Amazon, they have gone through stronger selective pressures, and they might have already be locally adapted to these more seasonal and extreme or drier climates, for example. And together with other studies comparing population level vulnerability and physiology, we believe that there is a lot of susceptibility to local extinction, even in a very common and high dispersal species such as Kentropyx calcarata. So the next step that we had was to try on and map the patterns of climate adaptation across the species range and evaluate for, let's say, a possible center-periphery signal of climate adaptation along the Amazonian Cerrado ecotone, and if there might be a role for, let's say, evolutionary rescue between locally adapted populations. Just a quick water break. So to do that, we integrated genomic level data and niche modeling with landscape ecology to try and predict these range shifts and potential for evolutionary rescue. And the results I'm going to present next, they are actually part of a larger project in which we propose this integrative framework using different species with different ecologies. So Amazon savanna specialists, lowland rainforest specialists, and more generalist species. So today I'm going to present the results for Kentropyx calcarata. There are a work that is underway, but that we basically integrate genomic data, eco-environmental data, and also predictions of future climate change and land use and deforestation in South America. So to do that study, we used RedSeq data for Kentropyx calcarata, the forest specialist, in order to conduct the genome environmental association analysis, as I defined it earlier, and search for candidate SNPs likely under environmental selection, while also accounting for the neutral underlying genetic variation. So we then started off by inferring the neutral genetic population structure, right? And we used two different methods to check for concordance. And they both agree on recovering 5 main clusters, which are roughly delimited by the Amazonian river basin, which agrees with the results from Cronenberg work with mitochondria and protein coding genes. And then we moved on to carry the GEA analysis to identify the signatures of climate-driven genetic variation, based on these associations between allele frequencies and local climates. So we investigate a range of environmental variables. In the end, we selected the ones that we hypothesized to be more important to our system and were not overly correlated. And we used two sets of approaches to control for rates of false positive that are known to occur in this type of analysis. So the first set of analysis was a redundance analysis that enables for simultaneous detection of candidate SNPs across multiple environmental predictors. So they basically show us how groups of SNPs can covary in the multivariate environmental space and are known to be able to detect even weak associations. And in this case, to control for the neutral population structure, we use the PCA axis as covariates. So the RDA, the redundancy analysis, recovered 334 candidate SNPs under selection for this system. So the second set of analysis that we use was the latent factor mixed models, or the LFMMs, which screens each of these SNPs at a time for signatures of local adaptation. And at these scans, it uses the neutral genetic structure, the K value, as a latent unobserved variable. So that's a form to control for the neutral variation. So different from the RDA, the LFMM, it's run one at a time, one of these SNPs at a time. And using this approach, we recover 124 candidate SNPs under environmental selection. So for the downstream analysis that I will show next, we just focus on the SNPs, on the candidate SNPs that were recovered by both methods, which was 10 SNPs. So then looking at those SNPs, we classified climate adapted genotypes according to the predominant climate in which they occur. So here you see the plotted distribution of the multi-locus genotype for all those 10 candidate SNPs for all individuals, for one individual, and here for all of them. And we could verify that the environmental space across the distribution of the species can sort of be divided into a wet or less or non-seasonal environment, and also a drier or more seasonal environments here in Eastern Amazonia, mostly along the ecotones of the Amazon. So therefore, we classify individuals as potentially adapted to wetter and non-seasonal climates, or drier and seasonal climates, and also a few ones as intermediate genotypes. So the next step in our approach, we want to model the distribution of individuals with genotypes that are likely adapted to different climatic conditions across the whole species range. So for this, we use ecological niche models, and we also use dispersal simulations that were constrained by dispersal barriers. So that's a forest specialist species. So deforestation, non-forest areas, they actually were going to act as a barrier for this species. So then that's what we did. We used projections of future deforestation, which likely are underestimated given the current scenario that we have, but still they model two different scenarios, one that is kind of more pessimistic and a more extreme one, right? And then we kind of predicted the future ranges of each class of locally adapted genotype and compare that with the whole species range. So we could sort of map whether each of these classes would be able to track new environments, which are the yellow areas, and suffer the range shifts, or if they would likely become locally extinct, or the red areas in the figure, or if they could just persist in situ, where the environments haven't changed, which is the blues here. So here we have the whole results cross individuals, the wet adapted and the dry seasonal adapted across both scenarios, the pessimistic and even more the extreme scenarios of land use. And as we can see, if for the wet adapted genotypes, there is quite a lot of areas predicted to be stable or even allow for range expansion. For the dry seasonal genotypes, we mostly recover very high levels of potential local extinction, habitat loss and local extinction across the entire range. So they would likely retain just very small patches of suitable areas, this in 2100, in both scenarios, both the pessimistic and the extreme one. So from this modeling, we then want to estimate whether there is any potential of evolutionary rescue between these two locally adapted genotypes, as that would be inferred as areas in 2100 where one of the genotypes overlaps with areas where the other one is predicted to lose their range. So that would be equivalent to say that there is a potential of spreading climate adapted genetic variation in the future across populations. So that's sort of the framework. So, and what we found, so this is the result, and what actually we basically found no significant evolutionary rescue predicted from the dry genotypes into the wet adapted genotypes. And here is just the other way around, the results from the wet adapted genotypes rescuing locally adapted dry genotypes. And as we can see, if in the less extreme, which is a pessimistic scenario, but not as extreme, we actually can recover quite a lot of areas of potential for evolutionary rescue in this direction that I mentioned. But when we look at the extreme scenario in the right, the A2 scenario, we basically lose all potential for evolutionary rescue from locally adapted populations. So we see very large areas of extinction, predicted extinction from the current range, and just these small purple areas here in the northern range of these genotypes. Or in other words, the potential evolutionary rescue potential is drastically reduced and really concentrated in just small portions, indicating that there will likely be a lot of biodiversity erosion in the southern parts of the Amazonia and the transition to the Cerrado, which is what I said here. So that's kind of brings, like I was talking about narrowing the adaptive bridge, but maybe this is just a case of all this potential just lost, missing the boat of probably rescuing other populations due to very extreme scenarios of the synergies between climate change and deforestation. So just to summarize everything that I said until now, so basically our studies, they've shown that ecological and climatic factors, they can affect speciation patterns and phylogeographic patterns at distinct temporal and spatial scales. And when we look at the more geological and historical time scales, we've shown that besides vicariance, ecological dispersal and divergence, they were very likely important drivers of speciation and expansion of the natural distributions of the species and their niches. And we also shown that there is a lot of trait variation between these lineages, and that those lineages, they will very likely have different susceptibilities and adaptive potentials to climate change. And of course, if you want to contemplate this potential for evolutionary rescue, then conservation strategies should be thought to incorporate population connections, things that if we are only under extreme scenarios, likely are not likely to happen, which is the main summary of the last study that I shown, that these extreme scenarios of environmental change, they will very likely hamper any potential for evolutionary rescue among natural populations at this large Amazonia Cerrado environmental gradient, and cause very high erosion of biodiversity in such a diverse an amazing region. So as the future perspectives of this study, we want to increase inference and generalization power by investigating more species with distinct ecologies, distinct range distributions. So we started off with more broad distributions, but we want to look at more restricted range species as well, and different physiologies. And this way we'll be able to look if there are any signatures of convergence SNPs or genomic areas under environmental selections that could be triggering or helping this expansion and colonization of novel environments across species, across the community, right? And be able to map under this adaptive perspective, the winners and losers under changing environments at the community level. So I have some interesting studies results already for other groups that I couldn't show today, but that's sort of the future perspective to put this all together. And of course, we want to do functional genomic validation of our studies. And as I said, interspecific, yeah, it's the last one, Lara, just wrapping up. So the interspecific variation of thermal traits, extinction risks, and climate-adapted genomic variation, they are very likely to affect vulnerability to climate change and the assembly of communities and biodiversity patterns in the Neotropics, which is a very mega diverse and complex landscape, right? And phylogeography is then an emerging field that can help us bring together this understanding of neutral and adaptive process in shaping this whole dynamics. So with that, I finished. Thank you very much for everyone that is here for your attention in a Sunday and a Sunday afternoon. But I also would like to thank the organizations for the organizers for inviting me, and my lab, which are a big group of students and collaborators that are very present and participated in all the steps of the work, photographers that help the presentation stay beautiful, the scientific collections, and of course, all the funding agencies that support our work. Thank you very much.
No, thank you. A very interesting talk. Before moving to question, let me remind you all the audience that the other talk of Pineiro is going to start only in a few minutes, if not has already. So if you are interested to hear the questions and answer from Fernanda, please stay. If not, you can continue to the other talk. Okay, let's go with the questions. So the first one says, several works rely on genetic sample, but in a pandemic point of view, where the access to samples are limited or sometimes impossible to get, is possible to contribute with revision research in the field of bio and phylogeography? If yes, how?
If I understand, it's let me see, pandemic point of view. Well, indeed, it's been hard to conduct field work right now in the field. But as I said, here we have several institutions that carry collections with previously collected samples that under collaborations and etc. can always be taught. I'm not sure if I got Lisla's question exactly, but I think that's what she asked, right? So I think that for a study that at this size that I'm showing, it's something that we collected along a couple of decades. So that's not something that was collected in a few years. It would be very hard, logistically almost impossible to put together this dense sampling with just a couple of years. So that's something that builds up with lots of field in the ground, field work, collaborations. I mentioned that that's a very collaborative work. I have collaborators in several institutions. So that's mostly how we can cover ground right now in the Amazonia, which is very challenging to conduct all this research, right? So I guess,
I will
thank you, Lila, for the question.
I would also like to point that there are some databases, for example, GeneBank, where you can find a lot of information to make new research without going to the...
Yeah, probably not to detect signatures of selection, because then you need the genomic libraries and etc. But the phylogeography assessment, of course, you can always build up from GeneBank and everything else.
Yes. Okay,
Thanks,
the next question is says, How is the political panorama involving these studies? Policymakers worldwide are using them to support their decisions. Do you know some examples?
Well, okay. Well, Thiago, thanks for the question. So that's, I mean, here in Brazil, that's the challenge, right? To make the policymakers understand that population level processes, they are important for biodiversity conservation. Because there is still a lot of, oh, if not species level data to support the decisions tend to go past because people think, oh, that's just a population. If it's not here, there is there. But there is a variation of vulnerability and potential adaptation potential. So that's something that I'm very insistent. Every time I go with policymakers, I always make this point. So, but it's still starting off, right? So it's not something that it's at least in Brazil. But abroad, seen more applicable. Like in Europe, there are some studies, usually smaller geographic scales, right? That comes with landscape genetic type of approaches in smaller scales. But yes, you can actually. Genetic variation is a very important component of biodiversity and adaptive genetic variation as well. So that's the fight that I try to reinforce. Thanks, Thiago.
Okay, we have one more that says, how do you think fragmentation population size decrease could affect your potential for local adaptation and evolutionary rescue?
Thanks, Leander. Good to see you here. Thanks for the question. Well, it's a very important question. I mean, with genomic data, we sort of can try and estimate effective population sizes, and those can be included in the migration limitations that we place in the ecological model. So this is sort of the frontier because it's very intensive to do. And when you look at, because you also ask fragmentation at the landscape level, we do know that it's hard at this geographic scale that we are working to look at these border effects. And I mean, things are being deforested as we speak right now and in a very high rates. So those are very important aspects to be included in future studies. At this point, we started off like a higher scale. And I guess that for that refinement would be better started in more regional scales, I would say would be more feasible to a model in our models, including our models. Thanks, Neander.
Okay, I have one question. The mechanistic model that you developed, they were at species level or lineage level?
Lineage level. So we just plotted in the same figure, but they were conducted for each of, I mean, we had 4 out of the 10 lineage. We couldn't sample all of them, but for those 4, they were lineage level.
And how did you evaluate if they overestimated? Because that's the main critical thing that says to the lineage level models, if they overestimate or not the future impact.
Well, I guess that using the approach we didn't control for overestimation. That's right. But we have run just correlative models, and we noticed that the mechanistic models, they weren't overestimating, let's say, more than the correlative ones. And when we incorporated the genetic level, the standing adaptive genetic variation, we actually didn't overestimate it at all. So we estimated less suitable areas. So, but that's a good point. I mean, it's something that to keep in mind and try to control for that. And I guess that with physiological data, the tendency is not as overestimated as in the correlative models.
Yeah. And one last, because I'm interested in the direct development frog, Brachycephalida, have you had any works with this group, or do you know any work that tests the distribution of Brachycephalida or any species in relationship to the reproductive mode?
the small, which group? Sorry, I didn't. The brachycephals, the small.
Yeah.
I don't know, out of my mind. I don't know of any study that's doing that right now, but it would be very interesting because then they have a very like altitude. The range shift is going to be completely different. It's not in the latitude, but right instead in the altitudinal range. But I'm not sure if but there is a lot of people working on the brachycephalids here in Brazil, so there might be.
Okay, let me thank you.
Thank you, Lara. Thank you.
Thank you for your talk. And well, I invite you all again to the next talk. If you want to continue the talk or the discussion on Discord, please do so in the channel Lobby Icebreaker, sorry. And that will be all. Thank you very much to you and to all the audience. See you soon.
Thank you. Bye-bye. See you soon. Bye, Lara.